In the winter of 2008, a Russian archaeologist named Alexander Tsibankov was working through sediment at Denisova Cave in the Altai Mountains of Siberia. He handed a fragment of bone to the dig director. It was a small piece of a finger's distal phalanx, the last segment, the tip, so small it could balance on the pad of your thumb without you really noticing the weight. Unremarkable. Probably human. Filed as Denisova 3. Logged, bagged, kept cold. Nobody expected anything from it. 2 years later, Svante Pääbo's team at the Max Planck Institute for Evolutionary Anthropology in Leipzig extracted mitochondrial DNA from that fragment and ran it through a sequence comparison. The result came back and someone had to look at it twice. Then probably a third time. The mitochondrial DNA of Denisova 3 differed from modern humans by approximately 385 base pairs. For reference, and this reference matters, so hold on to it. The mitochondrial distance between us and Neanderthals is around 202 base pairs. That number, 202, used to feel like a lot, like a deep divide, like the outer edge of the human family. And then, 385 appeared on a screen in Leipzig, and the outer edge moved, and everything we thought we knew about who was in the world with us had to be rewritten. Not revised, rewritten, from a fragment the size of a grapeseed. Right. So here's the thing about the two-branch model of late human evolution. It was not a bad model. It was built on the best evidence available for most of a century, and that evidence was physical. Bone, stone, morphology, the shape of an eye socket, the angle of a chin, the architecture of a brow ridge. The story it told. Archaic humans and modern humans diverged. Modern humans left Africa. Neanderthals held Europe and Western Asia for hundreds of thousands of years. And when modern humans arrived, the Neanderthals disappeared. That was the whole story. And the reason it was the whole story is that the tools we were using, morphological analysis, comparative anatomy, stratigraphy, could only see what they were designed to see. They could see shape. They couldn't see sequence. Denisova Cave is worth a moment, because the cave itself is part of why this discovery was possible. It sits in the foothills of the Altai Mountains, about 150 kilometers south of Barnaul. The interior temperature holds at around 0 degrees Celsius year-round, cold enough to act as a natural preservation system. DNA degrades fast in warm environments. In Denisova Cave, it didn't. By the time the finger bone was found, archaeologists had established that the sediment layers represented a continuous record of occupation spanning at least 125,000 years. Multiple layers, multiple species, an archive of biological material that most sites on the planet simply cannot provide. Denisova 3 was morphologically undiagnostic, meaning, looking at the bone, you cannot tell what kind of human it came from. Under the old toolkit, it would have been catalogued as probably modern human and moved on. But Matthias Meyer, working in the Leipzig lab, had refined a method called single-stranded library preparation for ancient DNA extraction. Standard preparation loses the shortest, most degraded fragments. Ancient DNA is short and degraded. Single-stranded library preparation captures those fragments. The result applied to Denisova 3 was a nuclear genome sequenced to 30-fold coverage from less than 10 milligrams of bone. This grapeseed-sized scrap of a finger yielded a genome of higher quality than a hospital would run on a living patient. And what that genome showed required building an entirely new category. The nuclear genome placed Denisova 3 in a population that had split from the lineage, leading to Neanderthals approximately 400,000 years ago. Not from modern humans, from Neanderthals, a sister group sharing a common ancestor with them after both had already diverged from us. 3 branches, not 2. And the third one had been in the record the whole time. In sediment layers, in museum drawers, in bone fragments catalogued as something they weren't. And the method simply hadn't been able to see it. Here's what that means practically. If you spend a century sorting fossils by morphology and encounter a population that doesn't look dramatically different from its neighbors, you file it in the nearest available category. You will not create a new branch of the tree because the method gives you shapes, not sequences. The genome said the box was wrong. Now the nuclear genome did something else. It showed that the Denisovans had interbred with modern humans, not occasionally, but in a way that left measurable percentages of Denisovan DNA in living human genomes today. And the distribution is geographically structured in a way that tells you something about where the contact happened. Melanesians, the indigenous peoples of Papua New Guinea, the Solomon Islands, Vanuatu, carry between 4 and 6% Denisovan DNA. David Reich at Harvard published the key analysis in 2010 in Nature. 4–6%. That is not contamination. That is biological inheritance from a population we didn't know existed 2 years prior. And here is the geographical paradox. The finger bone is from Siberia. Melanesia is thousands of kilometers to the south and east, across open ocean, which means the Denisovans were not a small, isolated Siberian population. They were spread across a vast swathe of Asia, and at some point, modern humans moving through that territory met them, and then kept going, carrying something with them. If you're still here after all that, you already know what kind of channel this is. Subscribe, because this is what we do every single week. The research that doesn't make it to a museum panel for another decade. The findings that require you to build a new box before you can put anything in it. Right. Deni. In 2012, the excavation team recovered a bone fragment from layer 11 of the cave, designated Denisova 11. Run through collagen fingerprinting first, a technique called ZooMS, zooarchaeology by mass spectrometry, which can identify species from protein signatures without extracting any DNA, flagged as hominin. Then the mitochondrial sequence came back Denisovan. Then the nuclear genome came back, and Vivian Sloan, also in Pebo's lab, had to look at the results several times. Denisova 11 had a Denisovan father and a Neanderthal mother. First generation, not a distant admixture. The actual mother was Neanderthal. The actual father was Denisovan. This person, known now as Deni, was the direct biological child of 2 different species of human, published in Nature in 2018. And that's the part that breaks the model. This is a cave with fewer than 24 individuals sequenced. The sample is tiny, and in that tiny sample, there is a first-generation hybrid. You don't find the exception in a sample this small if the exception is actually exceptional. You find it because the meeting was normal. Because Neanderthals and Denisovans were crossing paths in the Altai with enough frequency that producing children together was, on the scale of decades and centuries, not unusual. The cave was a crossroads, which somehow makes it stranger, not simpler. Go one level deeper into Deni's genome, and it gets even more specific. Her Neanderthal mother was genetically closer to Neanderthals later found in Western Europe than to those recovered from the same cave. A migration within the Neanderthal range encoded in one child's genome, and her Denisovan father carried traces of Neanderthal ancestry from hundreds of generations earlier. A different contact event, a different time and place. One bone chip, multiple waves of migration, contact and mixing across thousands of years. A biography of a species written in a single individual. And that individual was sitting in a box with thousands of other unidentified fragments until someone ran the collagen analysis, which is its own kind of thing to sit with. Look, the Tibetan Plateau. Elevation 4,000 to 5,000 meters. Oxygen partial pressure roughly 40% lower than at sea level. Tibetan Highlanders carry a variant of a gene called EPAS1, involved in the cellular response to low oxygen. That is dramatically different from the variant in Han Chinese populations at lower elevation. Rasmus Nielsen at Berkeley identified this as one of the strongest signals of recent natural selection in the human genome. The variant prevents the overproduction of hemoglobin that causes blood to thicken at altitude. It is, in measurable physiological terms, the difference between thriving at 4,000 meters and struggling to function. When the team ran that variant through genomic databases in 2014, looking for the closest match, it matched the Denisovan genome. Not Han Chinese, not any modern human population outside Tibet and adjacent highland groups. The Denisovan genome. The interbreeding happened, by current estimates, around 48,000 years ago. The variant drifted to low frequency at lower elevations and was kept at high frequency on the plateau, where natural selection made it essentially obligatory. Now roughly 80 to 90% of Tibetan Highlanders carry it, a 32.7 kilobase segment of a gene that diverged from our lineage 400,000 years ago is, today, allowing millions of people to live in one of the most physiologically hostile environments on Earth. The past is not past. It is running through blood. The Aita Magbukon people of the Philippines carry approximately 5% Denisovan ancestry, one of the highest proportions in any living population. Per al 2021 current biology study, Aboriginal Australians carry between 4 and 6% alongside their Neanderthal contribution. And David Reich's lab found evidence of a second distinct Denisovan admixture event in East and Southeast Asian populations, a different Denisovan population, a different time and place. Not one meeting. At minimum two. The Denisovans had their own internal structure, their own migrations, their own divergences within a range we are still mapping. And there is the sediment. In 2021, Elena Zavala and colleagues at the Max Planck Institute published results in Nature from 728 sediment samples at Denisova Cave. Ancient hominin DNA extracted not from bone, from the dirt itself. 24% of those samples contained ancient human DNA, a genetic occupancy map built from soil. The cave remembers, chemically, everyone who was ever in it. The sediment record shows Denisovans and Neanderthals occupying the cave across overlapping time periods, which, combined with Deni, tells you the cave was genuinely shared by two different human populations who were also, at some frequency, producing children together. Now, the Harbin Cranium. In 2021, a team led by Chang Ji at Hebei Go University described a large archaic skull recovered from the Songhua River in northeastern China, hidden at the bottom of a well during Japanese occupation in the 1930s. Forgotten, then rediscovered and donated to science in 2018, named Homo longi, the Dragon Man. Several researchers have proposed it represents a Denisovan. The genome has not been extracted. The case is currently contested in good faith. The uncertainty is real. What is not contested is the Jahi jawbone, found in 1908 in a Tibetan Buddhist monastery in Gansu province, subjected to ancient protein analysis by Frieda Welker and Jean-Jacques Hublin's group in 2019, published in Nature. The collagen fingerprint placed the individual in the Denisovan clade with high confidence. At least 160,000 years old, found at 3,080 meters elevation. Denisovans were on the Tibetan plateau long before the modern humans who would inherit their EPAS-1 variant arrived. They had been there long enough to adapt. And then they passed something on, and there is the deeper layer, the super-archaic contribution. Denisovan genomes carry a signal of ancestry from a population that split from the lineage leading to all three species, Denisovans, Neanderthals, and modern humans, more than 1 million years ago. Homo erectus, or something in that grade, is the current best candidate, roughly 4% of the Denisovan genome, which means the Denisovans themselves inherited biology from a lineage older than any of us, and passed some fraction of that forward. A chain of biological transmission across more than 1 million years, still traceable in living genomes. The method cannot yet identify the source precisely. We have no sequenced Homo erectus genome. But the signal is there. The ghost is in the machine. Before we close, drop a comment and tell me which part of this hit hardest. The finger bone, Deni, the EPAS-1 gene. The question at the end, I genuinely want to know, here is what I keep returning to. The Denisovans were not hidden because they were rare. The genetic evidence places them from Siberia to the Tibetan Plateau to Southeast Asia to island Melanesia. This was a geographically expansive, internally diverse, evolutionarily successful group of humans who occupied much of Asia for hundreds of thousands of years. And we didn't know, because the method could not see them, not because the bones weren't there, because the tools were reading the wrong signal. Shape instead of sequence, anatomy instead of genome. We have now sequenced 3 individual Denisovans from one cave, collagen evidence from one jawbone, sediment DNA, a genetic footprint across a third of the globe, and almost nothing else. No confirmed skull, no skeleton. The Denisovans occupied Asia for hundreds of thousands of years and left us 3 sequenced individuals and a jaw, which means there are sites being excavated right now where Denisovan fossils may already have been found, catalogued under a different name, and shelved. Nobody ran the genome because nobody knew there was a genome to run. Pabo, in his Nobel lecture in 2022, put it simply, the tools of ancient genomics transformed what we can see in the fossil record. Not by changing the fossils, but by changing our ability to read them. The fossils were always there. We just needed a different kind of eye. The question is, what else is sitting in a box right now, in a museum repository? Labeled archaic human, undiagnosed, waiting for someone to grind off 10 milligrams and run it through a cold room in Leipzig. What is the next grape seed? What is the next branch of the tree that we drew with only 2 branches because that's all we could see? The Denisovans were never absent from the story. They were absent from our ability to read it. And if that's true of them, then the question of what else we're missing is not philosophical. It's methodological. And the answer is still being written. In labs processing sediment and extracting nanogram quantities of degraded DNA from bones catalogued 60 years ago as something they were not. The story of the Denisovans is not a closed chapter. It's a proof of concept, proof that the archive is larger than the index, and proof that the most important discoveries in paleoanthropology right now may not be happening at excavation sites in the field. They may be happening in the drawers we already have.